Okay, so Vardhan and Prerana, there are about 11 open questions in the Q&A window. So if you could just read out the question and the person asking the same and give the answer, y'all can take it one by one basis, your subject matter expertise.
Sure, thank you. So I'll take this question. Can we also use the type of algae present in coral for identification? So based on my understanding, this algae is microscopic many times, and one has to, of course, dissect and get the algae out of coral tissue. As you are aware, corals are protected in India under Schedule 1 of the Indian Wildlife Protection Act, which means that you are not allowed to tamper or handle corals. So the answer is that can you use type of algae present in corals for identification? You can. There are different types of zooxanthellae or this algae dinoflagellate. To identify algae, yes, you can use that, but to identify coral, so it's not that certain zooxanthellae will select certain type of corals. So its answer to this is no, it won't be accurate, and also perhaps no need to, because the coral morphology, if you look at the structure, you will be able to identify. I hope I answered that question. So I'll move on to next, or do you want to, Prerana, do you want to take next question, which is some question about fish?
Vardhan, let's do one thing. Let's just go through the questions as they're upvoted. So if they're all coral first, no problem, you take it, and then we can move on to the next one.
All right. Okay. Yeah, so the next question is, how can we distinguish between corals and sponges? So this answer is fairly simple. As I explained in the presentation, there are these coralites, what you need to see is the shape and the structure of coral colonies, or if you are seeing something that looks like coral, you have to see that. Now sponges, they have, because they are filter feeders, they have those spicules and they are very characteristics, and you cannot mistake, though they look like round, sometimes from far they will look like corallites, but if you look at them, the structure is different, and because they are filter feeders, you will see that they are porous in nature. Whereas coral, because it's hard coral, which is made up of calcium carbonate, the structure itself will be different. So it is easy to distinguish, but again, it all comes with experience, and there are some soft coral in particular, such as acrophyta, they are confusing with hard corals. But again, this, when you see the sponges and corals, you will be able to differentiate. You can also look at the pictures and you will be able to differentiate based on spicules. I will read the next question, which is, Vardhan, do you need some, this is instructors to assist you on your trip to Angria Bank next time you go there? Why not? The answer is why not? It will be really nice to get experience divers and marine biologists as a team to go and explore. But the area of Angria Bank is more than almost 2,000 square kilometer. What we were able to explore was very limited, and we did 66 dives, but that is very as a group. So it's very little. So yeah, as in everyone is welcome. We can talk this later. Next, SSI is going to answer this. Next is how these algae arrange themselves into these polyp structures. Do they convert themselves into a tissue? Yeah, so there is no, so this algae, zooxanthe algae, is free flowing, and there is a mutualistic relationship between algae and coral polyps. So coral polyps or these zooxanthellae they have symbiotic or mutualistic relationship. Coral polyps produce carbon dioxide and water as byproducts of their cellular respiration system. And the zooxanthellae cell use carbon dioxide and water to carry out photosynthesis. And this is how this mutualistic relationship works. There is no, it's not that they, in their coral stomach tissue, they are present. And because they are microscopic, one cannot say that they are, I really don't know how they attach. the next question. How do new coral species get formed and what characteristics characterizes a new species? So as Prerana mentioned, as biologists, we use new species. So first thing I would like to clarify, coral taxonomy is easy, but it is easy up to genus level. When it gets to species level, it is very difficult. And what characterizes a new species, one has to do molecular analysis, and how coral new species are formed. As I explained, that they have all forms of reproductive strategies, which means that they have asexual budding. They also have hybrids, and because they are broadcast spawners, they are known to every now and then they are known to produce new species. And how this happens is because when these gametes are released in the water, they are planktonic, and these larvae are known to float. And because oceans are open systems, so in our waters, very likely we will, because of ocean currents, we will get larvae from other areas. And when these gametes or these eggs, when they fuse, and that's how the new species are formed. But again, identifying corals to species level is tricky, and one has to collect specimen, identify and see it. So even we identify as ecologists, because what we are really interested is the functional role of these corals, or as well as fish. So we don't go to up to genus, up to species level. We go up to genus level, and that is also a good one. Yeah, the next one, how long do corals take to build themselves? This is so it all depends, and different corals have different life history traits. So there are some corals such as acropora. They are very fast growing, and they grow very fast, and they after attaching within 2 months you will start seeing coral recruit or young coral. Whereas there are some corals such as those Favia, Montastria, or all these massive boulder corals, they are very slow growing, and they take years. So when you see a small colony, and another interesting part is that by just looking at size, you cannot tell how old the coral is. There are some corals which are very small in size, but they're as old as, such as brain corals, what we commonly call as. They are very, sometimes they are very old. They are as old as 100-year-old. And how they build is, of course, by the process of photosynthesis, and they grow in photic zone, which is where light penetrates, and using photosynthesis and those zooxanthellae, they form their calcium carbonate structure. And that's how when there's a reef accretion, which is loss, and accumulation, which is there's a term called reef accretion, which is when calcium carbonate formation and the loss, and that equation is called coral accumulation, and it's a continuous biogenic process, and it keeps happening, and that's how the reef is built. Is there a good course in by Rupi? Is there a good course in marine biology, ecology in India? A degree or postgraduate course shall also be okay if you can recommend a good one. So my answer to this is that there are good courses in India, but it all depends, because as a field biologist and as a marine biologist, if you study in textbook, or if you have bookish knowledge, it's of no use. It's important to go out, and if you are a student, it is important to intern and to get field experience. There are some good courses. There's course in NCPS, which is in wildlife sciences, which is good. There are WIA course, and then there are a few specific courses such as in Goa University, then Karwar, and there are many good courses. But then at the end of the day, what you learn entirely depends on your own effort and your experience, and how much you put in field. And most of knowledge and learning happens while you are in field. At what stage zooxanthellae form a symbiotic association with coral? So as soon as they lodge themselves into any hard substratum, there is a mutualistic relationship that forms as soon as they lodge themselves, and that's how they start growing, in fact. Then the next question, which is how these algae arrange themselves into these polyp structures. Do they convert themselves? I think I answered this question. Yeah. Next question. Can you suggest any reference material for identifying juvenile fish? Again, I think Prerana will answer. Prerana, do you want to take some questions?
So there's this question, which apps could be used to identify fishes? So there are a number of apps, and so there's Fish Snap, which you just have to upload a photo of a fish and it'll help you identify fishes. But also on like Facebook and everything, there are many groups which can also help you identify a number of fishes. So mostly the fish apps, there are a few, like there's this Neek, Fish, NEAQ website, which has a really good interface for identifying reef fishes. But I think that most of the fish apps till now are for identifying game fish. So not like from a research or anything in that point of view. So that was there. And can you suggest any reference material for identifying juvenile fishes? So I refer to this book especially for identifying juvenile fishes. It's called The World Atlas of Marine Fishes, which has a lot of juvenile stages of different types of fish. But apart from that, I think quite a few of them, like the Helmet Debelius reef fish identification, all these books, they have, you have Grismeck's Animal Life Encyclopedia that also has a lot of juvenile fish identification. So yeah, that's that. Which is the best place to dive around India to see these beautiful species of fishes and corals? I think many parts around India are really beautiful. You'll get to see a different type of habitat. If you go to see Andamans, it'll be a different experience. In Lakshadweep, since those are atolls, so you'll have a lot of beautiful clear blue sand lagoon areas. That's also there in Andamans also. Or even, as a matter of fact, Goa has some really nice places to dive, a really different kind of experience. But yeah, that's also really beautiful. So I think, I think in most of the coast, mostly I think the western coast of India and the Gulf of Mannar would have really nice places to dive.
Yeah, so to add on, of course, Andaman and Lakshadweep they are one of the best because of the visibility.
Yeah.
But in India, Andaman Nicobar are most diverse because it is believed the coral originated. One theory says that in Southeast Asia, and because Nicobar is just 190 kilometers from Indonesia, from the Coral Triangle, the biodiversity is very high in Andaman Nicobar Islands, followed by Lakshadweep, and again Gulf of Mannar is also biodiversity hotspot. It is exceptional. Some parts are exceptional in terms of biodiversity. And then Gulf of Kutch. What you have along the west coast and some parts of east coast are patchy corals. But then, of course, there are these surprising reefs like Angria Bang, which are, of course, different ballgame. But yeah, so all these sites are very good to dive.
Then the most voted question is, how can a diver start a career in marine conservation, and what skills are expected of him or her? I think mostly it's passion, and then the fact that you want to know more and more about whatever topic is about marine life. Marine conservation also, yeah, I think mostly it's passion. Yeah, Vardhan, would you also like to elaborate something?
Yeah, you are right.
What
You said. I would just say what is needed is right mindset, because
Yeah.
At the end of the day, if you have right mindset and if you really want to conserve then you can. And when, of course, degree or knowledge in the field, of course, helps. But if you are a diver and you are interested, you can always learn and master your skills. You can perhaps do a course, or you can work at dive centers and start marine conservation, because marine conservation is an activity. It's you keep doing it. So the skill sets are very broad. I'll take
Absolutely.
next question. How do new, no, that is done. What corals feed on? Oh, this is an interesting question. So the corals feed on by catching tiny floating animals called zooplanktons. So at night, coral polyps usually come out of their skeleton to feed on, stretching their long, those stinging tentacles that you see to capture all the critters and small planktons. Whatever the prey that they feed on, they're pulled into the polyps mouths and digested in their mouth. That is one way that they feed on. In addition, coral polyps, they also use sunlight to make sugar for energy. This energy is then transformed to polyps, providing much needed nourishment to the coral. And in return, coral polyp provides algae the carbon dioxide. So it's both zooplankton as well as that algae that live within the coral polyps called zooxanthellae. That's how they feed on. Next question by Siddharth Raju. I have been told that coral bleaching occurs due to the algae being unable to continue the process of photosynthesis. If the algae receives CO2 and H2O from the coral and receives sunlight as usual, exactly what happens to the algae during the temperature change that prevents them from being able to photosynthesize? So I guess this is a comment more than question. So this is the answer, is that because this algae is very sensitive to temperature changes, when there is also 1 or 2 degree rise, they get expelled out, and that's why the bleaching happens. So while you are right that bleaching occurs due to algae being unable to continue the process of synthesis, it's mainly it happens mainly because they are not able to withstand the temperature changes, and because of that they expel out of tissue. I hope I have answered this. Next is, do the bleached coral return back to life and grow? Yes, coral, when the bleaching happens, at the peak of bleaching, coral, you'll see many colonies of white bleached coral. But then if bleaching is prolonged, that is if the temperature is prolonged, then corals die, and what you see, the surface which is white, gets turned into small turf algae starts growing, and then many times macroalgae starts growing, and then it's a dead tissue. Otherwise, if it's not prolonged perhaps, and many times corals turn pale, not completely bleach. And when if the temperature drops down, if the conditions are conducive, then corals again come to life and starts growing. Do the fishes consume? So interestingly, in 2016 bleaching, at the peak of bleaching, the previous question, at the peak of bleaching, almost 50% corals had bleached, but the mortality was only 5%. So it all depends on what is the extent and how long the bleaching or the temperature rises. Then there are a few fish related questions by Anil and Ashwin. Do you want to take those, Prerana?
Yeah. Do fishes consume coral rubble? So, well, so coral rubble is basically like fragments of corals which have been broken. And if they've been broken for a long time, then algae starts forming on them. So basically that is food for a lot of fishes. So yes, fishes I've seen fish feed on coral rubble. Vardhan, would you like to elaborate on that?
No, you have answered, yeah.
Yeah. And then the next question is, how do new coral species get formed? No, what's the purpose of? So, as I mentioned, the purpose of schooling of fishes is basically, it's like how, as kids, like some people say that stay in a group to avoid the bully or something like that. So schooling of fishes is basically also is like uh, to escape predation. Like if they are in one group, then mostly the predator will not eat them. Or it also, if they are in a group, it also, if the predator eats on a part of that school of fish, there is still some chances of that group of fishes to survive. So there are many different strategies why fish school. Uh, what is the reason behind synchronized movement within the schooling? So I know that a lot of these fishes, they form different shapes of schools to sometimes even scare the predator. So all these fishes come together to unite to look like a big fish to basically scare off their predator. So the reason so that could be the reason behind the synchronous movement within schooling fishes. Uh,
Yeah, also just to add on, there are two terms that are used. There is shoaling and there is schooling. And it's important to understand that because it's a evolutionary strategy that is used to reduce predation. And so in shoaling, a group of fish stay together by maintaining for social distance, are shoaling. And if a group is swimming in the same direction, then it is schooling. Means these are loose terms that are used and sometimes they're used interconnectedly, but this is so just to repeat, when they're moving in one direction, it is commonly called as schooling, whereas shoaling is they stay together for for social reasons. Yeah. Go on, Prerna.
yeah. So how do new coral species get formed? I think these are all coral. There's MS microbiology.
Yeah, I can take this.
Yeah, Vardhan, you take the questions that are at the top four right now. I think they're all related to corals.
Okay. Uh, I think I answered this. How do new species get formed and what characterizes a new species? I answered this question. Then there is a question by Aishwarya. I'm a MSc microbiology student and very much interested in coral ecology conservation and resilience programs. So can you suggest me the courses which I can pursue in this lockdown period so I can uh, pursue in these programs? Uh, I guess you are attending this session and thanks to SSI for conducting this ecology course related courses, and this is the right place. Plus Venkat, he is also doing series of talks, so there are many good talks that are happening, and many researchers and conservationists as well as enthusiasts are giving talks. So I think that is one good way to go. And there are no, as I understand, there are no courses as such, but just listening to talk and understanding and doing self-study will be useful. That's what I would say. Then there is a question by Karishma 9. How big a coral can grow? Um, I even now this question I think I have answered, but corals can grow as big as 5 to 6 meters. So there are some corals in Middle Button Island and which are mainly Porites species, which are really big. So this submassive and massive, especially Biflostria or Porites, they grow huge. Then there are of course plate corals. They also grow big, but the max that they can grow is around 5 to 6 meters. Um, uh, and of course, there are some corals which are as small as microscopic, and you can't even see them. Then there's a question by Karishma. Do they require certain pH for their growth? Yes, they require certain pH. And there is a whole new field of studies on ocean acidification. So because of climate change and because of changes that are happening globally due to pollution and range of things, what is being said is that sea is getting more acidic, and as a result, so when we say that sea is getting acidic, that means the pH of the sea is changing, and when that happens, the especially giant clams or all the clams or the most reef building corals as well as associate, they have limestone, and that gets brittle. So that is having a huge impact. And yes, they require a certain pH level. Next question is, can coral be planted to grow in case of coral bleaching event in an area? Coral restoration doesn't work like plant restoration. So while it is tricky because you can only grow corals, like you can plant corals such as acropora, because they branch off, and you can take that branch and tie it on some iron frame and it will grow. But then it is very difficult to plant a coral to grow them in the case of bleaching event. Also, it requires a lot of money, time, energy, and effort. And even if you do that, you will be able to restore very small area. So it is tricky. And of course, there are people who are working on building super corals and corals which are resilient and who can tolerate high temperatures. But then the success rate of all these efforts are very limited, and it is a difficult task. Then the next question is by Aditya. I'm pursuing my master's degree in marine biology in Karwar. I'm interested in corals and want to learn more about corals and get into coral taxonomy. And to start with, I want to work on the same for my dissertation. So what would you suggest to get on and start with? I would suggest you are pursuing your master's. As soon as you finish your master's, and if you are interested in corals and taxonomy, I think it's good that you approach people who are working in field in the field and who are studying corals as well as working in the field of marine conservation. You can intern with them. You can, if there are positions available, you can work with them, and that will be good. Your question is that you want to work on for your dissertation. Perhaps you can, if you are interested, you can write to us and we can help you with that. Then there is another question by Aditya, which is, in the process of coral bleaching, zooxanthellae are expelled out by polyps. So what is actually happening in the process? Does the algae produce some chemical compounds which is disturbing for polyps, due to which they will want to get rid of algae or any other things? So you're right. Chemicals do play a huge role. In fact, there are some interesting studies that coral, when there is a broad scale spawning, if there is a parent coral, even the larvae tend to attach nearby areas, and that happens due to chemical cues. There are different clades in zooxanthellae, and yes, there are chemical compounds they play a huge role. Next question: what is the difference between corals that are found in intertidal region and in underwater? So this is a question by Ashwin, so the main difference of this is, if you see in intertidal areas, corals are exposed to several extreme events, such as there is a lot of wave action, there's a lot of because those corals get exposed, there are lots of what is the word, the disturbances that they go through, and they are very, because of that, they are very sturdy and hardy species. But then there are corals that are found in intertidal. The same corals are also found in deeper water. It's not that there is any difference in terms of their morphology, structure. Only the difference is how they adapt to the environment. So what I would say is that the corals which are in intertidal, they're well adapted to survive in that condition as compared to corals which are found in deeper water. Next question is by Sharwani. Prerana, do you want to take this question?
Yes. So how does a large school of fish manage to conduct synchronized movements? Is it by some form of communication? So schooling of large schools of fish stay together. Yes, there is a combination of really good communication between fishes and their movements. And one of the reasons why, I think there's still a lot of research to be done on how and why these fishes move together. But there's one, so fishes are always giving cues and receiving them. So they're very sensitive because they have sense organs which is like, they have the lateral line. So these lateral line pick up they're very sensitive and pick up cues from the environment, like the pressure right beside them, if there's a fish or not. And that sort of helps them maneuver and move together. So I think that's how they achieve this synchronous movement. Yeah. And then when coral is bleached, what the reef fishes do they find some other healthy coral to inhibit, or they live within the bleached coral? So corals and reef fishes, as we said, that they are so interdependent with each other. So if there are bleached corals, then the life around them also reduces. So yes, they do tend to look for other healthy corals to inhibit. Yeah, Vardhan, would you also like to add something?
Yeah, right. I would just like to talk about this whole schooling.
Yeah.
This is a very interesting study which talks about most schooling fish, they lose their ability to school in the dark. So that means that the visual senses play a very important uh, role. I'll just comment on that. And it indicates that vision is important. As I know that other sensors are also used, such as pheromones or sound, they also play an important role. And of course, there is, as you said, that there is supporting evidence. Also, interestingly, most schooling fish, they have some lateral lines, such as barracuda, which is running through each side of the fish from the gills to the tail. And in laboratory experiments, at least it is shown that these lines of when they are removed or when they place the marker, fish are not able to. So I think visual, pheromones, sound, all of these factors play important role, and people are still studying, and it is complex, but it is at the same time very beautiful and amazing phenomena which is used across, of course, even mammals such as dolphins, even they are known to and do this. Yeah, that's just a comment. Yeah.
Yeah. So there's another question, very interesting. Hi, Prerana, you said you can't fish in your studies, and what's the best method during a dive? Keeping in mind that they are mobile and you can miss out on counting. So I think, Akshita, you've answered the question yourself, that there are many methods to count fish in the water when you're diving or snorkeling or anything. But as you said, that it depends, you have to keep in mind why you want to count fish and what type of fish you're looking at. So there are some methods which are there are some methods which are good for fish that are mobile. There are there are certain methods for counting fish uh, that are pretty sedentary, like how Vardhan was doing in his studies. You have transects, uh, bell transects, or stationary point counts. So it all depends on what fish you're looking at, and also why you want to count.
Yeah, and just to add on, it depends on your question essentially.
Yeah.
And yes, there are chances of missing out, but then as
Yeah.
ecologists, what we really are trying to understand is uh, the functional group. For example, all these zooplanktivores, they are in thousands, such as fusiliers. So we don't count them as, we don't count when we are underwater with on a underwater paper. What we do is we estimate their numbers. So we know more than 100, more than 200, more than 500. And even when we do some analysis, when we understand that there are more than certain groups, more than 1,000, we know that what role they are playing. And that the idea is, because the question is to understand the functional group. So yeah, that's what it is. Yeah,
And the next question is, I'm studying public policy and I'm interested in coral and ocean conservation efforts. And what are the ways I can work with research organizations? I think many research organizations require people who can read in between the lines about government's policies on different things, like for example, you have a recent national fisheries policy, which has a lot of in between things which need to be studied. So there is a lot of uh, scope, and it is an important area of work. What are the ways I can work? Well, you'll just have to associate yourself, I think, with uh, organizations. Yeah.
Yeah, agree. And most organizations have policy wing. So you can just write to them and
Yeah.
introduce yourself and show your interest. And perhaps because there is always that disconnect, as Prerana mentioned, between biologists, researchers, and conservationists and policymakers. And they're often talking at each other. They're not talking to each other. So it's really important that more policy people are on board and they are joining conservation organization.
And then we have the last question. Yeah. I want to know the scientific name of black coral.
So my answer to this is I don't know. And also black coral is a very common term. And if you see in my presentation, I did not dwell into common names for a reason, because common names are very misleading. There are so many corals that are black in color. There are so many corals that are red in color. And these are common names. So if you can give some more specific in terms of their structure or something, then I would be able to help. But based on this question, my answer is I don't know. Yeah.
Okay. Thank you, Vardhan. Thank you, Prerana, for doing this. Guys, this is obviously a very expansive topic. Both coral ID and fish identification in themselves are really exhaustive subjects. And of course, there are going to be many more questions that you will have. Vardhan and Prerana have already shared your email addresses on the chat window. I hope that is okay. People were asking for it, to get in touch with you in case they want to ask certain questions. If there's any other suggestion that you have, or for all the listeners and all the attendees, we will be sending up a follow-up email with the webinar all over again as a replay, with all the reference points that they have used, all the links that they suggest that you should look into, and they will also share their contact details in that as well, including their email address. And then you can just get in touch with them for any questions that you might have post this session. Otherwise, thank you so much, everyone, for being a part of this. Again, Vardhan, Prerana, taking the time out to do this for us. I know you guys put in a lot of effort to do this, and we're really thankful. and hopefully, there are people now who know a little bit more about coral and fish identification than before this, and everyone starts taking their steps to making a difference to the environment. So guys, we will be ending the webinar now. Thank you so much. We will wait for about 2 minutes before we close down the webinar.